Welcome to the Science Whiz, the only game show where science is at the forefront. Today we have 5 contestants who will be representing their colleges. Firstly, we have Rina from the Committed School of Natural Sciences. Next, we have Kia from Excess Central College. Then Shanice from Quito University. Akili from Beta University, and Ria from Novo University. These contestants will be competing for the grand prize of an A in their biochemistry final exam. Today's topic will be lipid metabolism. Let's get right into the first segment. A series of questions will be asked based on diagrams, and at the end, 2 contestants will be eliminated. Right now, we're going to be starting with you. From the diagram, what is the name of the first step?
This step is called the committed step, where acetyl-CoA is converted to monomer-CoA.
Positive about this answer? Very good, very good. Correct. Kia, moving on to you. An enzyme and cofactor are required for this step. What are their names and what type of enzyme is it?
The cofactor required is biotin, and the name of the enzyme is acetyl-CoA carboxylase. This is an ABC enzyme. That means it requires ATP, biotin, and carbon dioxide to function. This enzyme takes the carbon from the carbon dioxide and adds it to acetyl-CoA to form malonyl-CoA.
On point, correct. Shanice, moving on to you. What features acetyl coenzyme carboxylase has which makes it accommodate this reaction?
Because it is so easy. Well, this enzyme has extra active sites where extracellular enzymes comes into the cell and binds it. That's it.
Precisely incorrect. I'm sorry, it's not correct. We'll be moving on to Akini. Akini, would you like me to repeat the question, or are you sure about your answer?
I got this.
Okay.
You see, it is a swinging arm mechanism with 2 active sites. First, we have the biotin carboxylase, which adds the carbon dioxide to the biotin. Then we have the trans carboxylase, which then transfers the carbon dioxide to the acetyl-CoA molecule to form malonyl-CoA. The lysine side group and the biotin carriers facilitate the swinging arm mechanism.
Shanice, does that ring a bell?
No.
Akini, your answer was correct. It's okay. We're moving on to Ria. Ria, why is acetyl coenzyme A converted into malonyl coenzyme A? So in the cells, you have acetyl coenzyme A being converted to malonyl coenzyme A because it is a precursor for fatty acid synthesis. Ria, superb. That is correct. Moving on to the next question. Contestants, please have a look at diagram number 2. Have a close look, and then I will be asking you a series of questions. Did we all have a look? Is everybody ready?
Yes.
Renal, we're starting with you again. In part 1 of the diagram, what process is occurring and what is the role of a glycerol 3-phosphate? Take your time.
You sure this is part of lipid metabolism?
This diagram is precise.
I have no idea.
That is not the answer. I'm sorry. Here, this has been passed on to you. Would you like me to repeat the question, or are you okay?
Right, I think I have this one. This process is glycolysis. This is where glucose is converted to pyruvate. Within this process, glycerol 3-phosphate is one of the intermediates formed. Glycerol 3-phosphate is dephosphorylated to glycerol, which is part of the, what is that called again? Triazide glycerol molecule. This molecule is used to make fatty acids.
That answer is correct. Even with your glitches, you recovered. It is correct. Shanice, In part 2 of the diagram, the citrate formed does not enter the citric acid cycle. Instead, it leaves the mitochondria to begin fatty acid synthesis. What causes this to happen? Shanice?
So the high energy charge in the cell inhibits the citrate from continuing along the citric acid cycle. Yeah, that's it. So glycolysis is used to produce large amounts of energy for the cell. However, if the cell has enough energy, then the citrate doesn't need to continue along the TCA cycle to produce more energy. Instead, it leaves the cycle, enters the cytosol in preparation for fatty acid synthesis, where potential energy can be stored as stacks. Yeah, that's it. That should be clear.
Okay, Shanice, obviously you know this part. Yes, that was the correct answer. Moving on, Achille. In part 3 of the diagram, coenzyme A is converted to malonate but then converted back into coenzyme A. This seems unnecessary. However, why is it done?
Well, you see, to someone who doesn't know what is going on, it would seem unnecessary, but actually, you see, fatty acid synthesis occurs in the cytosol, and acetyl-CoA is necessary for fatty acid. It's a precursor for fatty acid synthesis. So therefore, the acetyl-CoA in the mitochondria has to leave the mitochondria and then enter the cytosol. But seeing as it can't permeate the membrane, what happens is that the acetyl-CoA is converted to malonate, which can then exit the mitochondria into the cytosol. Once it has reached the cytosol, what happens is that it's reconverted to acetyl-CoA and then the process is allowed to continue.
You have correctly proved that it was necessary. Now, part 4 of the diagram shows the committed step and the fatty acid synthesis. Please tell me, what is the committed step and what reaction does the enzyme fatty acid synthase catalyze? So the committed step is basically a non-reversible rate regulatory step where once a substrate enters, it is obligated to carry on throughout the end. Now, fatty acid synthase, on the other hand, is a multifunctional dimeric enzyme that catalyzes the process of fatty acid synthesis. Now, with fatty acid synthesis, it catalyzes cycles that involve amino coenzyme A, acetyl coenzyme A, and acyl carrier protein, and NADP, where we have the fatty acid being elongated by 2 carbons each cycle. In these cycles, you have 4 reactions that are basically the same. First, we're going to start with the condensation reaction, where we have monomer coenzyme A, acetyl coenzyme A being added to an acyl carrier protein, and we have carbon dioxide being removed. In this cycle, we have acetyl coenzyme A being the substrate that only reacts in this particular cycle. Now we're going to move on to reduction, where we have monomer coenzyme A is added while we have NADPH being oxidized to NADP. Now we're going to go on to the dehydration cycle. We have monomer coenzyme A being added again, however this time as it says dehydration, so we have water being removed. Lastly, we have another reduction cycle, where this time NADPH alone is being oxidized to NADP. Notably, the elongation of fatty acid synthesis stops when we have palmitate being formed, which is a 16-carbon, fully saturated long-chain fatty acid. Further elongation and insertion of the double bond, that is the 2 carbons, this occurs in the endoplasmic reticulum and the mitochondria. However, it involves different enzymatic processes, and that's my answer. We have come to the end of segment 1, viewers. Renal, Shanice, your time on the show has come to its end. Please stand. Shanice, you have to. Please stand.
It's so hard. It's so hard for this.
Shanice, walk of shame, viewers. Walk of shame. It's okay. We have another quiz sometime. Viewers, stay tuned. When we come back, Kia, Akila, and Rhea will be put to the test yet again. Please stay tuned. And Welcome back to the Science Whiz. In segment 2, participants will be asked one question each based on the synthesis and degradation of fatty acids. However, the questions need to be accompanied by diagrams and equations. The participants who answer their question to the best will move on to the final round. After fatty acid synthesis, long-chain fatty acids are converted in the cytosol to acetyl coenzyme A derivative by long-chain acyl coenzyme A synthetase thiokinase. These molecules are then broken down to release energy when the body is in its fasting state. We're going to start off by asking Kia, for the beta oxidation of fatty acids, the carnitine shuttle is essential. With the aid of a diagram, please do explain this process.
Well, first of all, long-chain fatty acids are converted to their coenzyme A derivative by fatty acid synthetase. This molecule cannot enter the mitochondria, however, so it is converted from its coenzyme A derivative to fatty acid carnitine using carnitine, and this is done by the enzyme CAT1, also known as CPT1. This can now pass into the mitochondria, and this is reconverted to the fatty acid derivative that we want in the first place. And the carnitine is sent back across through transporter proteins, and this process is done by the enzyme CAT2, also known as CPT2.
Okay, Kia, that was indeed correct. Good job in using your diagram and your explanation. Correct. Akini, are you ready for your question?
Of course.
Very good then. After the long chain fatty acid enters into mitochondria, they are oxidized to release energy. Please do describe this process.
Well, this process entails 4 reactions, right? The fatty acid chain is reduced by 2 carbons for each cycle, right? Each cycle contains 4 reactions. The first reaction is entirely cleavage, which produces 1 acetyl group. Then the second reaction is an oxidative, which produces NADH2. The third is a hydration, and the fourth is another oxidation, which produces NADH. This process is repeated for saturated fatty acids at even length. There's a formula N2 minus 1. This formula is used to calculate the amount of cycles that will be necessary to completely break down an even chain fatty acid, N is the number of carbon atoms in the fatty acid chain. Each cycle produces 1 acetyl group, 1 FADH2 molecule, and 1 NADH molecule. These high-energy molecules are then oxidized by the citrate cycle or the oxidative phosphorylation cycle, which produces a lot of ATP, and that is what leads to the break. That is the benefit of the breakdown of these oxidation.
My dear Kini, is this your final answer?
I believe it is.
Okay then. However, you got the steps wrong, mixed up. What it is oxidation, then hydration, followed by a second oxidation, and lastly cleavage. So Kini, I'm sorry, but overall your answer was incorrect. I'm sorry. Rita, my dear girl, please compare fatty acid synthesis to fatty acid degradation. Okay, so in terms of the differences between fatty acid synthesis and fatty acid degradation, I have a really pretty simple table laid out here. First thing, I'm going to say that fatty acid synthesis takes place in the cytosol, while fatty acid degradation takes place in the mitochondria. Next, we're going to talk about the intermediates. Intermediates in fatty acid synthesis are covalently linked to sulfhydride groups of an acyl carrier protein. In comparison, in fatty acid degradation, we have intermediates are covalently attached to the sulfhydride groups of coenzyme A. Moving on, main enzymes in fatty acid synthesis are acetyl coenzyme carboxylase and fatty acid synthase. In comparison, in fatty acid dehydration, the main enzymes are carnitine, acetyltransferase 1, CPT1, and carnitine acetyltransferase CP2. Now, lastly, we're going to be talking about the reductants of fatty acid synthesis is NADPH, while in comparison in fatty acid degradation, the oxidants are NAD plus and FAD. And that concludes my answer. So Ria, that answer was indeed correct. Great job. Okay folks, we have come to the end of yet another round. Kia and Ria will be moving on for another set of questioning, whilst Akini, we're sorry, but it's time for a walk of shame. Welcome back, and it's time for segment 3. In this round, the contestants will ask each other questions. During the break, they were asked to prepare their own questions for each other. Ria will start off by asking Kia a question. Kia will answer the question and ask Ria a question of her own. However, if one of the contestants are unable to efficiently answer the question they are asked, the other contestants who ask the question will have to thus answer the question of their own. So we now go to the contestants. Right. So Kia, an individual with biochemistry deficiency was shown to produce fatty acids at a greater reduced rate as compared to someone who does not have the deficiency. This is due to which of the following? A. To low activity of the citrate lyase. B. To reduce activity of malic enzyme. C. Reduce activity of acetyltransferase. D. Defective acetylcarioprotein. Or E. Reduce ability to form malic coenzyme A. This is easy, kid girl. You can get it.
Kind of just want to know. But I don't choose C, yes? C.
C. No.
No?
No.
What is the answer?
This question is quite accurate and C does not do justice. I'm sorry. That means I will have to show you how it's done, key girl. Firstly, let me say, biotinidase is an enzyme required to remove the covalent bond biotin from proteins. In the absence of biotinidase enzymes, an individual can become deficient in biotin due to the lack of free biotin in the body. The formation of malic coenzyme A via acetylcoenzyme A carboxylase requires biotin as a cofactor. So the answer is E, reduce ability to form malic coenzyme A, not C, when in doubt, to reduce activity of acetyltransferase.
So Ria, my question for you now. A carnitine deficiency can occur in a number of ways. Secondary carnitine deficiencies can be distinguished from primary carnitine deficiencies by measuring which of the following in the blood? A. Fatty acids. B. Acylcarnitine. C. Lactic acid. D. Glucose. Or E. Ketone bodies.
Here, the answer is it's B, acylcarnitine. I say B because the primary carnitine deficiency is when carnitine is not present within the cells. However, secondary carnitine deficiency, this is where carnitine is sequestered in acylcarnitine form. Thus, elevated levels of acylcarnitine in the blood is more apt towards diagnosing secondary carnitine deficiencies in comparison to primary carnitine deficiencies. Does that answer the question?
This is correct. She got my A.